Welcome to this explainer. Have you ever looked at a person or an animal or even a plant and wondered, how does a microscopic, completely invisible set of instructions build the sheer complexity of a living, breathing organism? Well, today we're going to take our time to answer exactly that. We'll use a simple, intuitive analogy, an architectural blueprint, to really wrap our heads around the deeply complex science of heredity. Specifically, we're going to explore exactly why our visible traits don't always perfectly match our underlying DNA.
So let's just dive right into this fascinating question and decode the hidden rules governing life itself. When you look around the natural world, or, even at your own family members at the dinner table, you might just assume that what you see is a direct one-to-one reflection of an organism's genetics. You see brown eyes, you assume the genes say brown eyes, right? But biology is rarely that straightforward. There is this deeply intricate relationship between the code we carry and the organism we eventually become. And unraveling that relationship is exactly what we are here to do today.
Here is our roadmap for today. We'll be looking at genotype versus phenotype, passing down the blueprint, Ploidy, which is basically counting chromosome sets, altered counts and altered traits, environmental impacts on phenotype, and finally, a summary and some real-world applications. All right, let's start with part 1. Genotype versus phenotype, the blueprint versus the building. To really begin making sense of heredity, we absolutely must distinguish between the hidden genetic code and the actual physical organism it ultimately creates. Think of the genotype as an organism's specific genetic composition. It's the secret architectural blueprint made up of individual genes, or alleles. The phenotype, on the other hand, refers to the organism in its entirety. It's the final constructed building. And hey, it is incredibly important to remember that the phenotype doesn't just refer to observable physical traits like eye color or height. No, it includes literally all aspects of an organism's internal anatomy, its microscopic physiology, and even its behaviors. Take Gregor Mendel's classic experiments with peas, for example. This brilliantly illustrates our point. Notice how a plant with a homozygous dominant GG genotype and one with a heterozygous GG genotype both build the exact same green pod phenotype, because that dominant capital G allele entirely masks the recessive lowercase g allele. An organism's outward traits just do not always reveal its underlying genetic composition. This is a foundational rule of biology right here. Different genotypes can, and frequently do, produce the exact identical phenotype.
Moving right along to part 2. Passing down the blueprint, Mendel's peas.
So the crucial question now is understanding how these genetic instructions actually get passed from parent to offspring to create all these variations. Looking at Mendel's law of segregation, we see that sexual reproduction is this incredibly orderly mechanism for transmitting genetic material. During meiosis, organisms produce haploid gametes, like pollen in plants or eggs in animals, meaning they carry only half of the typical genetic load. The two alleles present in a parent segregate independently, so each individual gamete only carries one. Then, during fertilization, each offspring receives just one allele from each parent, randomly pairing them back together to create entirely new combinations. It's basically nature's way of shuffling a genetic deck of cards.
And here's the crazy part. That random pairing, it's highly predictable. If you cross two heterozygous parents, let's say two of those GG green pod plants we talked about, the resulting generations will exhibit a famous 3-to-1 phenotypic ratio. Out of 4 possible combinations, 3 offspring will inherit at least one dominant G and show the dominant green phenotype, while only one will inherit 2 lowercase g's and show the recessive yellow phenotype. This predictable masking effect is exactly why scientists use what's called a test cross. By breeding an unknown dominant phenotype with a pure homozygous recessive one, scientists can force those hidden recessive genes to reveal themselves. It's like a biological lie detector test that lets us accurately determine the invisible genotype.
Okay, part 3. Ploidy, counting chromosome sets. We have the blueprint, but now let's introduce a new term, ploidy. Ploidy is essentially how many full copies of this genetic instruction manual an organism keeps on site in its cells. Technically speaking, ploidy refers to the number of complete sets of chromosomes. Most eukaryotic organisms, and yes, that includes us humans, are diploid, which means 2. Our cells maintain 2 complete sets of chromosomes, inheriting 1 full set from our mother and 1 full set from our father. Maintaining this standard diploid number generation after generation is exactly what ensures genetic constancy within members of the same species.
Now get this. This is a genuinely surprising fact. Chromosome number tells you absolutely nothing about the complexity of an organism. Every single organism has a fixed number, sure, but the correlation stops right there. Human cells have 46 chromosomes, arranged in 23 pairs. Yet the turquoise kingfisher, which is a beautiful but relatively small bird, has a whopping 132 chromosomes. The multiplier doesn't equal superiority, it's simply the specific genetic dosage that particular organism evolved to require to function properly.
Part 4. Altered counts and altered traits when the multiplier changes. But what happens when things don't go to plan and that established dosage is disrupted? Well, sometimes the number of chromosome sets deviates from the standard diploid number, and that can drastically alter the resulting phenotype. To understand these deviations, we have to differentiate between two major genetic conditions. Polyploidy is when an organism possesses entirely extra sets of all its chromosomes. Think of it as copying the entire blueprint manual multiple times over, giving you 3N, 6N, or even 8N cells. Aneuploidy, on the other hand, is a much more targeted error. It occurs when a single specific chromosome is either missing, called a monosomy, or present in triplicate, called a trisomy.
The phenotypic outcomes of these two conditions couldn't be more wildly different. Polyploidy is actually incredibly common in the plant kingdom, and it plays a huge role in plant evolution. It naturally gives us the familiar, robust traits of our favorite foods. Bananas are triploid, wheat is hexaploid, and those huge, juicy strawberries you ate? They're octoploid. In general, polyploids appear quite normal and often even larger than their diploid counterparts. Aneuploidy, however, severely disrupts genetic balance. In humans, aneuploidy, like monosomy or trisomy, is estimated to cause 10 to 25% of all pregnancy losses. When organisms do survive aneuploidy, they exhibit a specific set of traits caused by the abnormal dose of just those few genes, Down syndrome being a well-known example of a human trisomy.
Which brings us to Part 5, environmental impacts on phenotype, nature versus nurture.
Now, as powerful as genetics are, the genetic blueprint and chromosome counts aren't the only factors at play here. The environment where the building is constructed matters immensely. Consider this beautiful quote from the biological texts. A single tree, locked into its inherited genotype, has leaves that vary in size, shape, and greenness, depending on their exposure to wind and sun. It's poetic, right? The phenotype is not a rigid outcome. Even identical twins, who share the exact same genetic sequence, can exhibit physical differences. This is because epigenetic effects, which are modifications that change gene expression without altering the DNA-based sequence itself, along with nutrition and an organism's unique environmental history, all interact with the genotype over time. The sun, the wind, the soil, all of these nurture the nature, ultimately finalizing the observable phenotype.
Finally, Part 6, summary and real-world applications. This table perfectly summarizes the relationship we've explored today. The genotype provides the core instructions, much like an architectural blueprint. The ploidy determines the specific dosage, or the number of copies of that blueprint kept on site in the cells. And finally, the phenotype is the final building, the observable organism that has been constructed from those instructions, but also continuously weathered, shaped, and finalized by its environment over time. So why does all this matter? Well, understanding this exact relationship, that carefully manipulating a genotype or adjusting a plant's ploidy can achieve a highly desired phenotype, is the factual basis for modern biotechnology. For instance, current scientific efforts are heavily focused on genetic engineering to ensure food safety and security in regions like Africa. By precisely adjusting the genetic blueprints of vital crops, scientists aim to build phenotypes that can withstand harsh, drought-prone environments, naturally resist invasive pests, and yield far more nutrition for growing populations. It's pretty amazing stuff.
And that leaves us with a final thought for you to chew on. We have moved from Gregor Mendel simply observing the phenotypes of pea plants in a monastery garden to entirely mapping the human genome to intentionally engineering crops for global survival. As our understanding of ploidy, epigenetics, and genotypes grows ever deeper, what new phenotypic realities might we be able to build next? Thank you so much for joining me on this explainer, and keep questioning the blueprints around you.